We have other subsets of neurons like this subset right here that expresses proenkephalin very specifically. And in fact, this appears to be one of these subsets that we just don't identify in rodent data or mouse data at all. This seems to be like a uniquely human subset of nociceptor. And we have another subset that we can identify, at least putatively, based on its relatively specific expression in a particular subtype in the mouse. So nicotinic alpha-3 receptor is relatively specifically expressed in silent nociceptors in mice. And we also identify a population of human neurons that specifically expresses this. So we presume that this is the silent nociceptor population in humans. And in fact, if we look at some of the pharmacology data from people like Barbara Namer that do microneurography in people, receptor expression in this subset of neurons actually matches very well with some of the in vivo pharmacology that they've done in patients and volunteers recording from these neurons. So we feel pretty confident about calling these different nociceptor subsets. And in fact, in Steve Davidson and Nick Riva's paper, they identified many of these same subsets. The one they had trouble identifying was the CLTMRs. We feel like we've identified those right here, and they express a lot of GFR alpha 2. So to kind of hone in on some of these populations, I'll talk about 3 of them briefly. Here's the putative silent nociceptors. So again, these express nicotinic receptor alpha 3. But they also express some other genes relatively specifically. So one that really caught our eyes is cholecystokinin, or CCK. So CCK is a neuropeptide. It's been shown to be released by neurons in many different circuits before. CCK receptors are expressed in the rodent spinal cord. We also know they're expressed in the human spinal cord. And this might be an important neurotransmitter that this particular subset of neurons uses to communicate with dorsal horn neurons, or it may also have some efferent function in the periphery. It's very specifically expressed in this population. We also identified, like I just mentioned on the previous slide, a nociceptor subset that we don't really see at all in other species. This is this proenkephalin positive subset of nociceptors. And this one, so proenkephalin is a neuropeptide as well. Obviously, it binds to opioid receptors. But we also see urocortin, which is a stress-related peptide transmitter that's pretty specifically found in this nociceptor population. So this was kind of a theme that we noticed when we looked across all of these different subsets, is we have what I think is the emergence of a kind of neuropeptide code for different subsets of sensory neurons. And I'll talk about at the end of the talk how this might really help us as we start to better understand different subsets of neurons in the human spinal cord, because one might imagine that these neurons that express these particular peptides might connect to neurons in the spinal cord that express receptors for these peptides. So the last one I'll talk about is the prurigen receptor enriched nociceptors that we identified in humans. And the reason that I want to talk about these is because this is another kind of theme that we saw across these different subsets. And that's that while in mice, this population is very specifically marked by expression of NPPB and the IL-31 receptor. These peptides and receptors are expressed by this population in humans, but they're also found in other populations in humans. So some of this kind of specificity of markers seems to break down, and we see what we think is kind of a little bit more polymodality between these. So, you know, while I'm sure, or I'm not sure, but I think you can say with a decent amount of confidence that this population of nociceptors is involved in itch. It's likely that some of these other nociceptor populations are also involved in itch, and they express peptides that we know might be involved in itch signaling, and receptors for that matter. So we did a lot of validation, trying to look using RNA-scope at whether we saw similar distributions of expression to what we saw in the Visium data. And in the paper, we went through a lot of these different genes that we looked at with RNA-scope. I'm not going to talk about that in a lot of detail, but I just want to make another point here, related to TRK, TRK-B, and TRK-C expression, although I'll just focus on TRK-A and TRK-B. So NTRK1 is the gene for TRK-A, NTRK2 is the gene for TRK-B. So broadly speaking, NTRK1 expression seems to mark almost all of the nociceptors in human, and NTRK2 or TRK-B expression seems to mark most of the A beta population in humans. What's interesting is, in the adult rodent, especially in the adult mouse, TRK-A expression is restricted to the peptidergic population. During early development, it's on in all the nociceptors, but it shuts off in the non-peptidergic population and persists in the peptidergic population. But in humans, that doesn't appear to happen. It seems like there's no shutoff of the TRK-A gene in that other population of nociceptors, and it's expressed into even late adulthood. And here again, so TRPV1 at the mRNA and protein level, we saw its expression in essentially all of the nociceptors. TRK-A we see in all of the nociceptors, and when we record from these neurons, all of the neurons that we recorded from responded to capsaicin with depolarization, and most of them fired action potentials in response to capsaicin. Now, our recordings are almost entirely done from smaller diameter neurons, so I'm sure there are neurons that don't respond to capsaicin that are in the A beta range, but we were recording mostly from neurons in the cell size of the C and A delta fiber range. And consistent with the RNA and protein expression, we see that all of them respond to capsaicin. So Ursula Indastiga in my lab, who's a postdoc, has gotten really interested in trying to understand why these transcriptional differences exist in the human DRG. And to that end, she started doing A2CPS on human DRGs from organ donors. A2CPS is a DNA sequencing method that looks at open chromatin within a tissue. So we eventually want to do single cell A2CPS, but right now we're doing bulk A2CPS. In A and B is just kind of showing you some quality checks for our sequencing. I'm not going to go into detail on this, but the sequencing quality on this looks really great. It really helps to get very fresh tissue recovered very soon after cross clamp, which is a major advantage of working with a local organ donor group. And Ursula has found some really fascinating things in looking at this data. So I'm going to just focus on NAV1.7, SCN9A, NAV1.9, and TRPV1. So I'll start with TRPV1. So obviously there's a big species difference in TRPV1, and what we've been able to identify in the A2CPS data is that there are some upstream transcription factor binding sites in the TRPV1 gene in humans that we just don't find in rodents. And they're from very common transcription factors for Jun and STAT1. So this might be one of the major reasons that so many human nociceptors express these genes throughout life, or this particular gene throughout life. And there's open chromatin in the areas of these transcription factors, suggesting that they would be accessible to transcriptional regulation. And we see similar things in NAV1.7 and NAV1.9. So for instance, for NAV1.7, we see EGR1 transcription factor binding site in an open chromatin area. This is interesting because EGR1 is upregulated in chronic pain samples from the work that we've been doing with Pat Doherty. So suggests that this transcription factor might be involved in regulating NAV1.7 expression in humans in persistent pain states. So from our sequencing data, our spatial sequencing data, we can also ask some other types of questions. So we had 8 samples that we used initially for the spatial sequencing. 4 of them were male and 4 of them were female. So we also looked at sex differences. So the first question that Deanna asked when she looked at this data was the most simple one. Do we see any difference in the populations of sensory neuron subtypes between male and female organ donors? And that's what's shown here. And I hope it's very obvious to you that we don't see any differences. So there doesn't seem to be any difference in the representation of different subtypes of sensory neurons between men and women. But what we did see was if we looked in individual subsets of neurons, that there were some differences here. So this was one of the more interesting ones, and we also validated this using RNA-scope. So what we observed was in the putative itch nociceptors that the CGRP, calc-A gene expression, was about double the level in women versus men. So I don't know if this has a functional consequence on CGRP playing a role in itch or a greater role in itch in women versus men, but it certainly is a testable hypothesis. And it's also quite consistent with a whole bunch of studies that have now been published about big differences in CGRP signaling between men and or male and female rodents. Also, I saw Mike Hildebrand is on the thing today. He has a really great paper that just came out in Brain showing sex differences in BDNF signaling in the human spinal cord. So shout out to him, and everybody should also check out that paper because it's I think really an important finding and demonstrates that some of these sex differences that we're finding in animal models are also well represented in humans. So I'm going to transition now away from the spatial sequencing data and talk quickly about the work that Pradipta has been doing on sequencing from human DRGs that we recover from chronic pain patients through this collaboration with Pat Doherty's lab, looking at thoracic vertebrectomy. This patient population has, most of them have cancer, and most of them have cancer that is metastasized to their vertebrae. And so for people that have this in their thoracic vertebrae, they're having a surgery to repair the vertebrae, remove the cancer. But the DRGs are in the way of where they're going to put in the metal rods. So the DRGs come out. And this allows us to phenotype these patients beforehand and see or ask if they have pain in different dermatomes that match the DRGs that we're going to take. And then that allows us to do electrophysiology and RNA sequencing. So the electrophysiology has led to some I think, really important findings. One of those is that if you record from human DRG neurons in a dish that are coming from a dermatome in which people have neuropathic pain, even days after these neurons have been in culture, you can see spontaneous activity. And not only do you see spontaneous activity, you see fluctuations in the resting membrane potential. And as Terry Walters and Pat just published in a really nice paper in Journal of Pain, this is one of these findings that actually is quite representative of what's seen in animal models, where you have this fluctuation in the resting membrane potential, and also these the spontaneous activity that occurs with neuropathic pain. So we see about 20 to 30% of the neurons that are dissociated, and then we record from in culture from thoracic vertebrectomy patients with pain show spontaneous activity. And we almost never see spontaneous activity in DRGs in which people did not have pain. So we think this is a pretty clear indication that spontaneous activity in the DRG is a driver of neuropathic pain. So what Pradipta has been doing for several years, and this paper is up on bioRxiv, and we're revising it to submit back to the journal we submitted it to. We see again some pretty major sex differences